Thank you very much for the kind invitation. It's a pleasure also, always to be here. You know, the topic is a little bit wide, but we are going to be short and tight. So particularly just to have you an idea of how it's going now with the genetic diagnosis of neuromuscular disorders. So I will take more about some advances and perspectives. I always show my team here because I never have time, because I always run out of time to present my team. So I present my team in the first slides so I don't have to just to leave time for the next speaker. So these are my disclosures. Close, like Frank Sinatra, no? Very close to the microphone. So this is a type of concept. The diagnosis of inherited neuromuscular disease is challenging due to their genetic and phenotypic variability. Traditionally, neurophysiology and histopathology were primarily used in the initial diagnostic approach of these conditions. You know, with advances in molecular diagnosis, the Sanger sequencing, you know, that's short parts of genes and gene by gene was used at, however, there was time-consuming and cost-intensive process. The advent and accessibility of next-generation sequencing, because this allowed to sequence a lot of fragments at the same time with a lot of readings, is a process that really revolutionized not only the field of neuromuscular disorder, but genetic disorders in general. Some common neuromuscular phenotypes may trigger genetic testing by the physicians. They include myopathic phenotypes, isolated, persistent, recurrent, high creatine kinase CK, abnormal neuromuscular junction transmission phenotypes, neuropathic or motor neuronopathic phenotypes, upper motor neuron phenotypes, and associated with syndromic and multi-system involvement. So we have plenty of opportunities to request a genetic testing for the patients that we suspect that has a neuromuscular condition. But, however, a number of neuromuscular diseases needs a specific genotyping methodologies, and NGS may be applied as a second step, not at a primary step. For example, I put here some examples: spinal muscular atrophy, muscular dystrophy of Duchenne, myotonic dystrophy, and facioscapulohumeral. So you cannot ask specifically for an NGS test for these diseases as the first tier of study. Most of the remaining conditions can be approached with different strategies, such as disease panels, partial exome or whole exome sequencing, or whole genome sequencing. The broad range of applications opens new and more affordable possibilities to study numerous cellular processes at a single base resolution that this allows the NGS. However, there is a production of massive amounts of data that presents significant challenge for data storage, distribution, analysis, and interpretations. And finally, we have to consider ethical issues about incidental findings or actionable mutations that they may appear in this type of analysis. I'm going to be through more or less these things. We started with specific genotyping tests. During several decades, we were aware that we have to study particularly one gene or particularly one part of the gene. Now we are moving in this direction. We are with the panels or exome by NGS. We have plenty and different places just to analyze and to study. And the three more interesting things is that how we face, you know, the genetic diagnosis when we have a condition that we suspect is a neuromuscular disease, and we have to study that with NGS. And it's not one of the conditions that I mentioned before. So here we started with the panel that is targeted for specific genes. So if we know a condition that can be caused by more than one gene, and there is a specific phenotype, perhaps the panel is the best solution. However, when we suspect that something is wide above the phenotype, or the phenotype is not really the same, perhaps this is not enough. We will need more. And the second thing is, okay, we can study with exome. The exome can be clinical exome, when you say all the genes that are already known that cause pathology, or can be a whole exome, that means all the sequences that are transcribed from genes to proteins. And, sorry, can I go back? Okay. Oh, I have to finish this. So the capture of exome contain more than 85% of mutations related with genetic diseases. And if we have a phenotype that maybe is not complete for being into the panel of genes, perhaps this will be the next step, just to go directly to the exome, and this defined as enough that could be the right amount in a reasonable way to approach the disease. And finally, if this is not the way in which we can reach any mutations, we can go to the genome sequencing, the whole genome sequences, and we're going to see now what will be these circles means with all these colors. That initially is too much, is more than we need to approach a specific diagnosis, and so far can be used as a second step after the genome analysis if the phenotype is just a little bit wider for the panel analysis. And this is what I show you. When you see the amount of protein coding genes is only 2% in comparison with all the things that are really in the rest of the genome. And the possibility that the rest of the genome can give us an answer is much, much lesser if we approach directly the exome analysis. So this is another way in which we can show what is the exome in relationship with the rest of the genome. And of course, we know that the exome is the part of the genome made up of exons, sequences that we transcribed, remind we did a mature RNA, after the introns are removed by RNA splicing and contribute to the final protein product encoded by the gene. And for exome, it's not like, you go to the grocery and just ask for exome for a patient, because we don't know what happens with the patients. So we need a specific receipt just to include the exome in an analysis, because the best way we indicate the exome, the best way that we can interpret the exome and the utility of this tool will be probably higher. First, what we see in the patients, that is the phenotype. And we have to include the phenotype with specific numbers, the human phenotype ontology. I will be after with that. What we know about the genes that may be involved in that phenotype, having a good methodology that allows us to capture the exome data, because you have to select specifically that region from the genome and arrange an appropriate bioinformatic analysis strategy. That means a pipeline. And of course, talking about HPOs, this is the way in which with HPO, somebody in other country having that this is the HPO 4998, and we know that it's bowel femurs, so somebody in other part of the world will know that 4998, we are talking about the same problem in the patients. And remember, a methodology can help us to have approach to the diagnosis, but we need to have a really, particular approach in seeing the patients, and not figure out that because there are some mustache here that will be mustache all over the families. We need to analyze and we need to hierarch the clinical work of all of us, because the patient is the best book, the best data, the best interpretation. So remember, exome, genome, panels are only collaborators of the big work of the clinicians. And of course, once we have everything done and we have all the study done, how we identify the variants. We have genomic variants that we have to select depending on population variants and depending on the inheritance patterns that we are looking in the genealogy. That can be, autosomal dominant, recessive, or even just X-links, the genes involved, the previous clinical evidence, the type of variant that we are going to comment a little bit, and the validation and segregation in the family. And this is the way in which we can just really have a variant. They can be pathogenic, clearly pathogenic, likely pathogenic. They can be of uncertain significance, likely benign or benign. And how we can just put this one into the different lights that we see here. We should see the frequency in the populations, the evolutionary conservations, co-segregation in the family, functional studies, and prediction by bioinformatics programs. So we have the diagnosis variants that is helpful to provide categorical or probability of molecular diagnosis, and is more clear if this variant were previously reported with pathology in the different database like ClinVar, Decipher, or MIM. So we need a guide just to clarify if the variant is involved or not. However, most of the time we are going to have with new alternatives that we can call this uncertain significance, because they are not present in controls or in the literature that relates the variant with pathology. They can be in candidate genes, but pathogenicity depends only from prediction, not clear evidence. So that means that we need additional studies of co-segregation in the family and functional analysis. And of course, when we have a variant of uncertain significance, we can put that in a waiting list. What we call waiting list is put on hold until they change or they will new patients reported with this variant all over the world. So that's the importance of being connected with all the rest of the databases to include this variant in one category or in another. But there is another thing that is important when we analyze, the exome besides the panels. That is, there is incidental findings. Incidental findings means that you have, because of the HPO that is related to the patients, you find or you select a gene and you find a mutation in that gene that is not the real cause of the disease, but they can be related with another problem, for example, cancer, or can be also a carrier of other conditions that are very frequent in the population, and they will have a risk of having a baby with this problem in the future. And the next one is to define as secondary findings. This is an important difference because an incidental finding is something related that you have to analyze because of the HPO of the genotype and the phenotype of the patients. But secondary findings is a list of actionable genes that is published by the American College of Medical Genetics. In this moment, there are more than 75 genes that can be actionable. Most of the genes are related with cancer and with cardiopathies. That means that these genes can be actionable, and when you get this data, you can see the family and you can tell you, besides this problem, that is a neuromuscular problem, you have also a large QT syndrome. And why is important that? Because if you detect that in an asymptomatic family and there is not showing this phenotype in the family, they will help you to avoid future problems in this family. That's the reason that they call this actionable genes. And this list is updated, in different versions. And it's important to take into account when we do that we have to explain that when we indicate the study to the patients. That's what we call here consideration pretest. Just to give the information that what is going to be analyzed in the actual knowledge. Which variants are we going to report? The utility of the methodology, which means limitation and managing expectation, because when you explain to a patient that you are going to do an exam, that you can see all the genes on your body, so this really increases the expectation of the family and the patients. And of course, we need to have an informed consent that can be related with the research projects or a clinical application daily in the routine analysis. This is more important. It's not an indication to the patients, do this analysis and come back with the analysis. We have to explain clearly all these pretest considerations. And of course, depending on what we are doing to do, we need to explain them which genes, how they are analyzed, how to interpret, and how you report results. This is very important from our side. And finally, we do the post-test in which we disclose the results, and depending in the patients is related with the pregnancy or in the future reproduction of the family, you will have prenatal decision of informed relative to the pregnancy. I really suggest to read these guidelines for diagnosis next generation sequencing that is really explaining particularly the 3 issues that I explained here for you to go ahead with the explanation. So means that we are here in the process in genetic diagnosis by the NGS and can be particularly applied to neuromuscular disorders. When you have a patient, you have a clinical diagnosis, the informed consent to do NGS studies, and depending on the phenotype of the patient, we are going to do panels, exome, genome. We will have the interpretation. If we got a pathogenic or likely pathogenic variant, we will do a diagnosis confirmation and genetic counseling. And of course, if we have a benign or likely benign or a boost, we have to put on hold with the boost, and we should inform the incidental or actionable mutation, depending if the patients want or don't want to know that. So we have performed a survey in our 800 patients that we perform exome analysis in our clinics, and more than 90% of the families, they want to know the secondary findings that we can do in an exome analysis because they want to prevent other diseases in the family. This is a very important issue and have to be taken into account because depending on how they really request this information, we should disclose all that information after when we do the post-test visit with the information of the studies. So that means that for interpretation, we need a multidisciplinary team in which we have particularly the clinical orientation and the phenotypic data, the analysis and interpretation of genome data, and the genetic counseling and the family data. And we use all these sources of information to get a definitive diagnosis, and that can help us just to look for and to put a variant on hold or to give the variant the validation that is really pathogenic and is the cause of the disease in this family. And that's what we do. It's not a fact that you have only one person. You have clinical and laboratory specialists according to disease manifestation and clinics. We met with neurologists, we met with neurosurgeons, depending on that, clinical geneticists, genetic and genomic counselors, molecular biologists, and bioinformaticians. It's really important to know that there is also possible omissions of molecular or functional pathology of NGS in the clinical practice. For example, the exome analysis of the NGS cannot really detect the triplet expansion of the dynamic mutations. You will not indicate for a myotonic dystrophy, for example, an exome analysis. You have some deletions. For example, if you have a patient with Duchenne or you have a patient with Charcot-Marie, you are not going to indicate an exome analysis, even though now the pipelines of analysis of the copy number variations of duplication and deletion is improving. But you have a specific test that you can do with that. There is an important issue also of pseudogenes because the nature of the NGS studies, that is short readings that you are putting together, means that there are some pseudogenes that can have some mutations that they are really masking what's happening in the real genes of the disease. And this is the same with genes with high homology. And of course, the NGS does not detect the structural rearrangements. We need other tools to do the structural rearrangements. It's not the first time that we have a patient with a neuromuscular phenotype. They have a panel negative. They have exome studies and they are negative. And when you do a chromosomal study and there is a translocations that it is broken one specific gene that we have not seen with the previous studies. So take in mind that there is no tool or no method that replaces the other one. It's a collaboration between all the methods that will help us to reach the final diagnosis of the patients. And of course, they will not give information about what's happened with epigenetic regulation. If we have a gene that has no problem of structure, has no problem of variation, but for any reason is not active or is methylated, they will not appear in the NGS studies. A number of NDS diseases, I'm going to take this last minutes to specific talk about 3 pathologies: spinal muscular atrophy, DMD, and facioscapulohumeral, that need special attentions beside the NGS, because all the other pathologies, most of the other pathologies, can be really approached with that. What is our role in doing that? We have to decide which is the best tool and not ask, with the help of the geneticists, with the help of the multidisciplinary team, and not ask first and after do what we do with the analysis, okay? It's a combination decision that we have to do before we up the expectation of the patients. So, SMA is a compendium of genetics. It's have a determinant gene and have a modifier gene. Most of you know that this homozygous deletion of these genes will cause the disease, and all the patients have the modifier gene that is pretty homologous, pretty close, and is present in all patients. And the number of copies may vary from 1 to 6, and depending on that, you will have a specific phenotype. And this is what you see in the general population. There appears SMN1 and SMN2. There are some part of the population has no SMN2 but have SMN1, and both genes are located in the lower arm of the chromosome 5. What is important is the patients have always at least 1 copy of SMN2 and they are absent of SMN1. And there is no people just reported without SMN1 or SMN2, meaning that the SMN protein, even lower amounts of SMN protein, are necessary in all the cells. We don't have time to explain something about carriers. I will take the question after if you want. And of course, I want just to mention that the homology between SMN1 and SMN2 is just this cytosine 4TB that makes the difference. No? This is the definition of SMN1 and this is the definition of SMN2. But all these are different between SMN1 and SMN2. And we will see later that that can be related with the ability of this exon 7 to be included or not in the final RNA. So even though this is a definition of SMN1 and this is a definition of SMN2, you know, there are a lot of variation between patients with all this sequence that we have here. And of course, this change in the position A40 is a silent change that speaks, because TTC and TTT codify for the same, phenylalanine. However, the presence of the C attracts good proteins to include the exon 7 into the RNA, and the presence of a T attracts negative proteins to skip the exon 7 from the RNA. And that's what's happened. You know, the protein is complete when you have SMN1, and the protein is incomplete when you have SMN2. But however, when there is not SMN1, there is some amount of protein produced by the cells to defend itself and to produce some SMN protein, as we see here in the case of an SMA patient. And the genetic diagnosis is very straightforward, actually, because practically 95% of these cases can be detected by deletion of SMN1. And there is only 5% of the cases that can be detected, none can be detected by a quantitative analysis that could be 0, 1 copy, or 2 copies. What is important, if the clinical is very important, because if you have a patient that you suspect and you find the biallelic absence of SMN1, you confirm diagnosis. That's what's happened in the 95% of the cases. However, when you have a negative case that can be 1 copy or 2 copies of SMN1, this is particularly important when the population has consanguinity. Consanguinity can be related with rare mutations, and they could have really a patient that will lose the opportunity to treat because we think that it's negative for this study or that study. We will be later on that, and these are also point mutations that we have to look everywhere when we have a patient with suspicion. However, a couple of things about that. SMN1 and SMN2 is not always straightforward. First, it's mandatory for, particularly now with the age of treatment, both SMN1 alleles should be characterized. But in some patients, we have one allele and another is present, though the second allele cannot be characterized as mutations. These cases might be other neuromuscular or motor neuron disorders that might be concomitant carriers of SMA, because SMA has a carrier frequency between 1 in 40, 1 in 60. And we need to do a thorough clinical examination and additional genetic testing, including NGS of the SMN region and exome that can be performed to categorize these cases. What is the importance of that? Because if we treat the patients with an expensive medication, we thought that it's an SMA and really are treating an SMA carrier, and it has another disease that we cannot treat or we have no diagnosis. That's happened to us. We spent 10 years with the patients, with the patient that was a carrier, and say, okay, we have to look for the second mutation. And we never found the second mutations. And when we perform an exome analysis, another homozygous recessive disorder appears that was related that this is SMAR, SMA with respiratory distress. So we have this patient during 10 years thinking that was SMA. The family came to look for the treatment, and we say, no, we have to perform the exact diagnosis. We were looking for the second allele, and after they say, okay, perform an exome study, and the exome was clearly to clarify the diagnosis of these patients. So take this into account. The other thing is that the SMN2 copies may not always related with the clinical issue, because we see that most of the patients are severe, but not the other ones. And we see that there is a variant in the SMN2 that makes this exome could be more included. When we have 2 copies of SMN2 are always related with type 1 disease, but when we have this variant in the exon 7, that is C859GC, that opens a little bit more from the molecular point of view, the structure of the SMN2 interaction with the proteins, the patients will develop type 2 disease. But when we have 2 copies of SMN2 with the variant, the patients will be a walker. And you can imagine the newborn screening. In this case, you have a patient with 2 copies, but with this variant, we will see, oh, this is a typical type 1 case. We are going to treat immediately. However, with the variant, they will develop by natural history a type 2, or with 2 copies with the variant, they will develop by natural history a type 3 disease. So that happens in our 25% of cases. And a word about hybrid genes. That's what I mentioned, because this is a pure SMN1 gene and this is a pure SMN2 genes. However, patients may have mix of these things, and that means that perhaps they will make different ways in which the patients have the phenotype and different ways in which the patients can respond to medications. So this is under study, and I will be glad to have more results probably in the next conference next year with more data about that. So that's what is important, that beyond copy number variants in patients with SMA, we have to have in consideration a series of things related with quantity and with quality of the SMN2 genes. A couple of words in my last minutes of DMD and DMB. We know that Duchenne muscular dystrophy is an X-linked disorder caused by deletion, duplication, and point pathological variants in the dystrophin gene. Has no problems, but women will be affected because of X monosomy, translocation, X autosome, and by chance with 2 X chromosomes with deletion and mutations. I'm going to mention something that disappeared now, incidental finding in the prenatal context. I'm finished quickly, don't worry. Most of the cases that we, when do a prenatal studies, we perform CGH, array CGH in all the pregnancies. And we have that in the pregnancy there is also probes in the array CGH for the DMD, the dystrophin gene. What found, for example, in one family, that this, sorry? 2 minutes? 5. Oh, 5, thank you. So this is interesting. That's stress is like, you know, it's a car without gasoline. You know, no more. You cannot go further. Sorry. And that's what I want to show you. But that's an interesting thing. Because that family, nobody was aware of that, but she became pregnant, and we do the CGH analysis here, and a deletion on the dystrophin gene appears as a routine study of her pregnancy. And when we started to define, we see that this deletion was present in all these people in the family, and even in a 90-year-old grandmother with dilated cardiomyopathy. And this patient was unaffiliated myogenic and neurogenic signs without increase of CK, but had aneurysmal dilatation of the aortic root and the renal artery. So that means that we are facing now, because we are expanding the genetic of different disorders, we are having incidental finding of a neuromuscular disorder, and we have to study the family. And you see here in the fetus that there was an inframe deletion of the DMD gene, caused by all this generation was silent. A couple of words about facioscapulohumeral. You know that the facioscapulohumeral usually is very important because it's a contraction of the region here in chromosome 4, and there is always time consuming because we do Southern blotting and there is only in specific centers, and there have to be done. Now there is a new tool called optical mapping, and in the optical mapping you just label all the different fragments, and you know that it's a genome of reference, and thank you to the optical mapping, we can just really see that the ordering of these fragments will be different. And that's what we do. Validation, this has been published already, that the optical genome mapping can help really to do with the facioscapulohumeral diagnosis. We see here, for example, the allele size by Southern blot and the allele size by optical genome mapping are really corroborate the diagnosis. So we have a tool that can be really very interesting to analyze in the future with patients with facioscapulohumeral. And I finish my sequences talking about long-read sequences. We have been in these sequences of unique molecules. It's like you pass the molecule for a whole and it's reading each by each one. There are 2 methodologies. One is the OFOG nanopore technology sequencing that have ultra-long reads, commonly 50 to 100 kbs and up to 4 megabase, that is a really record, with an accuracy of 98%, and can have also methylation detection. That means that we can see when a gene is activated or inactivated, even though that has not changed in the structure. So, and there is the PacBio Hi-Fi sequencing that have also long reads, that have an accuracy of 99%, methylation detection. There are limited options for amplification-free targeted sequences. The thing is, here you pass for a whole, and here you make a circular amplification, and you read, and read it, what we say Hi-Fi read. So these will be probably tools that will help us in the future to study cases in which we don't find the mutations by typical methods. Even when we just get used to the exome, something new appears that we have to apply. Have in mind that optical mapping and long-read sequencing will be in the future valuable tools to help us in that. And that's what we are going to face the future. We're going to have a personal bank of genomic data. We will take genomic or genetic information relevant in the clinical context of the patient, and we will select genetic and genomic information to transmit to the patients. We have to be the ward guardians of that information and transmit to the patient when it's necessary. This is the future that we have. And remember that we can have genetic testing during life. We can do a genome analysis at birth and keep this information for all over, or we can have all these different alternatives during the whole life of testing each by each by each. In the future, we are going to have that information, but we have to grow up a lot with ethical information and the consideration for genetic counseling for the family. So I think I finished. Thank you for the 5 minutes, and I am open to question if there is time for any question. Thank you.
Thank you, Professor Dr. Eduardo, for this interesting subject. Any question at the end of the session? طبعا بنشكر الدكتورة ناجية على الدعوة الكريمة، وإن شاء الله يكون في ميزان حسناتك إن شاء الله في المجهود الكبير اللي أنت عملتيه إن شاء الله. طبعا بنشكر الجمعية للسبورت للنيوروماسكولار سبجكت. الحقيقة دور الجمعية واضح جدا في إنها تساعد الوحدات الصغيرة. طبعا دي حاجة كويسة. هتحب تسأل سؤال؟
Thank you, Dr. Eduardo, for your nice presentation. My question is regarding Duchenne muscle dystrophy and Becker muscle dystrophy. Can the genetic testing give us an idea about the prognosis, about the degree of deficiency, dystrophin, or we still need to go through immunohistochemistry?
This is a very interesting question, because even though there is the rule of Monaco that there is in-frame deletions will give a better prognosis than out-of-frame deletions, there is some cases that overlap, and depending on each family. For example, in the family that I saw you was an in-frame deletion, was a Becker muscular dystrophy, but there was a variant. Even there was a male in the family with problems in the school that were related to that variant. So I think that we should collect that, and so far, the golden standard that is MLPA analysis perhaps should be replaced with more particular information of CGHRI for DMD, and they will give us much better structure of what is really deleted in the gene. The important thing is that we have to know that it's a big gene, and MLPA gives only partial information that what is deleted, and we don't know what is really the long way to that. I have to. What's happened? No, no, because it's no something special. Because I move. That's what's happened. It's following me. Okay, so I think that that's still is a matter of progress, and we never take out, the clinical information, because there are some families in which the prognosis that you do with the genetic study will not be, even there will be some variability within the same family. So at this end, we still have to rely with some uncertainty when we go about prognosis of this, even though in most of the cases, this rule will apply. So I invite.
What about genetic testing through the life?
Yes.
How I go to the clinic, I want to do genetic testing, but I don't understand how this made it.
No, what I was mentioning is that you have, in the future, we are facing two alternatives. Now you have a tool that you can have genome analysis at birth, and you can keep that information.
Yes, yes.
And you can analyze depending on the necessity of the patient.
Yes.
Or you can have, all the testing around the life and a different testing that makes you different approaches for genetic counseling and for different tests. So I think that in the future, we're going to have genomic in health public, and we are going to have our own genome, keeping safe in one place, and anytime that we go to the physician, he will see, okay, let me see your genes, and let me see. If you're going to have a treatment, let's do the pharmacogenetics. If you are going to have a treatment for, or you're doing a preconceptional study, okay, let's see if you are a carrier of autosomal recessive disease, you know?
Yes.
That will be the way in which we are facing now the genomic, the public genome in the future.
Yes, yes. So at birth must be done genomic at birth for all people.
Exactly, and you keep. The problem
Okay.
is where you storage that and how you keep the information of millions of people,
Yes.
of millions of base pair.
Yes.
So storage data is still a challenge.
Thank you.